My paper was estimating the magnitude of near-membrane PDE4 activity in living cells. The paper was written by Wenquan Zin and Wei Feinstein and others, and it is presented by me. This paper was published in the American Journal of Physiology in 2015, and the authors are from different universities in a couple of different states, including Alabama, Carolina, Louisiana, and Texas. So some background on this paper is that PDE4 is a phosphodiesterase, which is a part of the cyclic nucleotide phosphodiesterase group. This means that they break the phosphodiesterase bond using secondary messengers such as CAMP. This shows that these PDEs are important regulators in signal transduction. PDE4 specifically uses CAMP. HEK293 cells are used throughout this experiment because they are human embryonic kidney cells. They are used because they grow and transfect really easily, and they have become the standard model for the study of kinetics and subcellular distribution. So it's important for this experiment where they are measuring the subcellular activity of the enzyme phosphodiesterase. So, throughout this experiment, they observed two populations of phosphodiesterase in the lysate of the HEK293 cells. Roloprem sensitive PDE, which is a specific inhibitor of PDE4, and 8-MM-IBMX, which is a somewhat selectively inhibitor of PDE1. Both Roloprem and 8-MM-IBMX are drugs that are used to inhibit this enzyme activity. IBMX, which is a generic form of 8-MM-IBMX, is a non-selective inhibitor of the phosphodiesterase molecules. The aim of this experiment is to determine the localized enzyme concentration within a subcellular compartment, specifically near the plasma membrane. So they're specifically looking for the activity of PDE4 near the membrane of the cell, as the title stated, and they were looking for the localized signaling complexes that exist within the subcellular compartments. They used this three-tiered approach, which is the measurement of, so first they measured the total cellular PDE activity in cell lysates and found the distinct PDE subtypes, as we already discussed, which were the Roloprem sensitive and the 8-MM-IBMX sensitive subtypes. They estimated the rate at which known concentrations of CAMP dialyze into the cells using plasma membrane localized cyclic nucleotide gated channels as the readout using patch clamps. And they utilized this information in a mathematical model that was published by Thomas Rich, one of the authors, to describe the activity and spatial distribution of these pharmacologically distinct PDEs based on these previous two steps. This data that is used to predict the enzyme activity based on whether they are using it as a one-compartment or two-compartment model. The first part of the experiment is used to measure the PDE activity within the cell at a specific volume. This then relates the volume and the activity. So it shows the total PDE activity with the circles, and the Roloprem sensitive, which is the PDE4 activity, and the 8-MM-IBMX sensitive PD activity are squares. This shows the decrease in volume has an increase in activity because it is becoming more concentrated. And so in reverse, a larger volume has a smaller activity of PDE. The activity of the enzyme by the volume of the compartment it was in was determined based on this set of data. And they were also able to determine inhibition constants that are then used in the math model to help them also determine the activity of the enzyme. In this first graph, it shows the increase in current as CAMP dialyzes into the cell. And then as roloprem is added, there is a large increase in current because more CAMP is being able to reach the cell membrane. In these last two graphs, it shows the same beginning portion as CAMP is dialyzing into the cell. Roloprem is added and there's a large increase in current. And then as 8-bromo cGMP is added, there is also a large increase in current because more cyclic is being brought near the membrane. And then in this final graph, it is a similar thing that just shows that 8-MM-IBMX doesn't really create a huge increase in the current of the cell membrane that is measured by these patch clamps. And then roloprem adds a large increase. And then IBMX at the end has little to no influx of current, increasing current. So, how does the data found support the model? The green line and the brown line in this first set of graphs. The brown line is what it would be if it was a one compartment model as determined by the mathematical model, and the green line is what it would be if it was two compartments. So obvious, and then this black line is the data that they found based on the certain cell conditions, because they wanted to determine if the cell PDE activity was a one compartment model or two, because a two compartment model would model that the PDE acted mostly in the cytosol, and that one compartment would be that PDE acted mostly near the membrane. And so that's mostly what they're measuring is PDE activity near the membrane, because they're trying to determine if PDE4 is specifically near the membrane or in the cytosol. And as it is seen, there is a the data fits more closely to the two compartment model. And in the second graph, the data is more consistent with a two compartment model because there is a clear and consistent right shift from the excised patch, which better fits the estimated data of a two compartment model than the inconsistent difference in data. The results of the experiment was that the near membrane PDE activity is 20-fold less than in the cytosol, which shows or suggests that the PDE4 follows more of a two compartment model because there is more of its activity in the cytosol. And the sensitivity of the model that they used to predict these parameters that suggests an accurate model to be able to fit their data to. However, the flux coefficient between the compartments depends on the volume of compartment, and the two compartment model suggests a single barrier between the cytosol and the membrane that is mediated by secondary messengers, which is unlikely because there are most likely multiple secondary messenger barriers. My discussion questions are: why is it important that PDE activity be more prominent in the cytosol than near the plasma membrane? And why is it unlikely that there is only one barrier mediated by secondary messenger molecules in a two-compartment